Switching gears, now instead of just mentioning structure, we're going to go through DNA replication. How do we make more of this DNA that we need? DNA replication is the process of making more DNA for new cells. Basically, we're copying, we're making duplicates. The replication process is the result of many different enzymes working together to separate the strands of existing DNA. I have to unzip this if I'm going to make copies. Copying one strand and producing a new daughter strand. Neither of the two daughter molecules is completely new. One strand of the new molecule of DNA is going to have half parent, half new, and the other is going to have half parent, half new. So if you look, blue is parent DNA. Coming down here where the copies are being made, I've got half of the parent, half of the new, half of the parent, half of the new. Now that we know what DNA replication is, let's go through each of these steps so that we can see how it works. The origin of replication is a short sequence rich in adenine and thymine bases that are held together by only 2 hydrogen bonds rather than 3, because the origin of replication is an AT-rich sequence. Less energy is required to separate the 2 strands than would be required if the origin were rich in guanine and cytosine. I'm going to go back for just a second. Going back, you can clearly see adenine and thymine have 2 bonds. Cytosine and guanine have 3. If we're talking about splitting this apart, it's easier to break 2 bonds than it is 3. Thus, having an AT-rich region means that it's easier to kind of unzip that area than it would be if I had 3 bonds. So when we talk about it being AT-rich, that's what we mean. We have this AT-rich area that's easier to unzip so that we can start looking at making a copy. During replication, during making a copy, topoisomerases unwind the DNA helix, giving access to another enzyme, helicases, unzipping enzymes, to bind to the double-stranded, this DS just means double-stranded, DNA at the origin. Remember, when we have DNA, it's all twisted up. I don't want it to stay twisted up. I need it to unwind so it's easier to unzip and see both sides of the parent strand. So topoisomerase is the one that unwinds it, makes it go straight, and then helicase you can think of as scissors. This is the thing that cuts and splits the 2 parent strands apart so that we have a template to follow to make another copy. Helicases break the hydrogen bonds, holding the 2 strands together, resulting in 2 separate strands. Even in the illustration here, helicase has a little pair of scissors because it breaks those bonds in the middle. The single-stranded binding proteins are going to keep the 2 strands apart. Here are the single-stranded binding proteins. The thing about the hydrogen bonds is they're kind of like magnets. They want to come back together. So if I just unzip and don't have something to kind of pull these things apart, it's just going to automatically go and come right back together. I don't know if you guys have ever seen the, I don't even know if I think they call it like a safety screen or something. And you walk out and it's just floppy screen material, but then there are magnets in the middle that and it seals the screen door so that you can go in and out without necessarily having to deal with a handle. DNA is kind of like that. If you don't have something to keep it apart, something that actually holds it apart, it would just go right back together like magnets. So our single-stranded binding proteins are pulling and keeping those two strands separated from each other. Remember that helicase just unzipped. There is directionality to reading DNA. I know that sounds weird, but it's true. DNA, I'm going to go back for a second. DNA has two spots that we label 5' and 3'. And it's based on the connection to our sugar. We have a hydroxide here and a phosphate. So the phosphate is where the 5' hookup is, and the hydroxide, which is OH minus, or OH in this case because it's connected right here, is the 3'. All right, so what does that mean? When reading a DNA strand, you want to go from 5' where the phosphate is to 3' where that OH is connected to the sugar, the deoxyribose in this case. So if I want to read DNA correctly, in other words, I want to read a book from the beginning to the end of a sentence, I go from 5' to 3' in direction. So this one, 5 is at the top, 3 is at the bottom. This is the correct strand to read from, again, the beginning to the end of a sentence. The other side is almost like a mirror image. So it would be reading the sentence from the last word to the first word. Wouldn't really make sense. So this strand is the backwards strand. It's going from 3' at the top here to 5' at the bottom here. Now you might be saying, why are you telling me that? Because in DNA replication, it's important to know which strand is in the right direction and which strand is in the wrong direction. Primarily because the way that you read that DNA, the sequence of events to read that piece of DNA and make your copy varies depending on whether you are the 5' to 3' strand or the 3 to the 5' strand. So I wanted to cover that real quick before we started talking about how this unzipping happens. Well, not how it happens, but how the replication happens. Knowing this information is important for that. All that information I just gave you about direction is pertinent to the next step in DNA replication. The enzyme DNA polymerase works in that 5' to 3' direction. It cannot work backwards. If you've ever seen information about records, like literal vinyl records, they work well in one direction, but if you were to twist the other way, it doesn't sound like music anymore. It just sounds like random noises. DNA polymerase works 5' to 3', and that's it. That's one of the reasons why I went through the direction of the DNA so much, because it's important to understand what that is so that you can understand how this works. All right, so zippers. When we unzip something, we usually go down so it splits from the top. I'm going to flip this image just for myself in the lecture. It won't be that way on your PowerPoint, but I'm hoping that it'll help you to kind of understand how this works. DNA polymerase 3. You hear the word polymerase, it means it's building something. If it's DNA polymerase, it's building DNA. It adds nucleotides in accordance with the template pattern. So remember, I have a parent or a template strand, and I've got the strand that I'm building. I've got the parent or the template strand and the strand that I'm building. So I have been unzipping from the top up here and coming down. This strand is the 5' to 3' strand. This is the one that DNA polymerase can read from beginning to end with no stops. It's equivalent to having, let's say, 5 sentences on a piece of paper and reading from beginning to end and it making sense. That direction for us reading is left to right, is the 5' to 3' for DNA polymerase. The other strand, this strand over here, is not in the right direction. So instead of being 5' to 3' here, this is 3' to 5'. This strand can't, since DNA polymerase can't go in this direction, literally I have to unzip a section and then it can come in and read from the 5' to the 3'. I unzip a little bit more and it can go in and read that next section from 5' to 3' until it gets to the section that was already completed. But it can't go the opposite direction. Because of that, because I'm having to make section by section by section as it unzips, we call this the lagging strand. And because this strand can go from beginning to end without any type of pause in between because it's in the right direction, we call this strand the leading strand. Now, DNA polymerase 3, which is right here, has been reading the template strand or the parent strand and adding the correct or corresponding or complementary partner. Just to kind of give you an idea, if this is my parent strand, I'm just going to make something up. Let's say the pattern is A, T, G, G, C, A, T, because that says cat, C, A, G. DNA polymerase looks at that and says, okay, I know that because of the whole A always pairs with T, meaning that T always pairs with A, and G always pairs with C, meaning that C always pairs with G. Because of those rules, I know when I see the parent, I know who to put across from it. So in this case, if I see an A, because of my rules, as DNA polymerase, I will put a nucleotide of T across from it. Same with T, I know A will go across from it. This started up here and is reading in this direction, and using this, G, C goes across from it, to add the nucleotides as I go. So just to fill in, if I have a T, it's an A. If I have a C, it's a G. If I have an A, it's a T. A, it's a T. G, it's a C. So I've used this as a guide to tell me who the complement is to make the daughter strand from the parent strand, or I use the template to make the new strand. In the case of the leading strand, as I said, as DNA polymerase 3 is going, it can just keep going because it's in the right direction. Here, however, leading strand. Because the other side is in the opposite orientation, it is termed the lagging strand, just so you don't think that I'm making up words. I only work in the one direction, so as this fork is opening, as it continues to unzip, I'm adding these pieces, literally from the right direction. All right. While the lagging strand is slower, I am still building both strands at the same time. But since the lagging strand has these little pieces that I'm making, I'm actually making, I'm making more work for myself. Each of these short fragments that DNA polymerase has made is called an Okazaki fragment. Right there. Each of these fragments that I have built, each individual fragment is not connected to its next door neighbor. There is another enzyme called DNA lyase, which you can see here as this green spiky looking thing, is necessary to kind of stick them together. So to ligate something, I've got two pieces, the cap and the marker. That's ligating. You're putting them together, you're gluing them together. In the case of the individual fragments, ligase comes in and glues them together. So that they're one continuous piece and not separate pieces. The ability to do this helps to make that lagging strand into a complete piece of, or new piece of DNA. Something that I didn't mention before, but is still true. When I want to replicate DNA, there has to be a jump start. I actually have to insert a little piece of RNA first, and then I can go through and finish with my DNA. Push comes to shove. I jump start with a length of RNA that is manufactured by another enzyme, RNA primase. Remember, if it ends in ase, it's an enzyme. And I said that we prime it, we start it. So RNA primase puts a little bit of RNA to begin the process of making DNA. Here, you can see a little piece of RNA that starts these strands of DNA being made by DNA polymerase. That's RNA primase. That's the enzyme that's putting that little chunk of RNA to start. In our lagging strand, it is very easy to see where this happens because, again, since I'm going backwards, I have to do a little chunk at a time. So there are a lot of RNA primings every time we expose one of those little pieces. However, in our leading strand, where we started, let's say, way back down here, that little chunk of RNA would be way down there. And because I'm not doing Okazaki fragments on the leading strand, I only have one little RNA primed area. In my lagging strand, you see it over and over and over again because, again, I'm going backwards. I'm going in the wrong direction. Instead of 5 to 3 prime, I'm going 3 to 5 prime. Wow, all of a sudden that sounded Australian, sorry. The RNA primer is not seen on the leading strand, like I said, because it's closer to the origin, which would be down below the board. But again, looking at the lagging strand, you can see how you've got those little pieces of RNA for every section that we start. Let's review the enzymes that are involved in DNA replication. Topoisomerase 1 and 2 uncoil the DNA and let it relax so that replication can begin. Topoisomerase unwinds so that we have 2 straight pieces that we can split apart. So both 1 and 2 do the same job. That's what they're in charge of. Helicase breaks the hydrogen bonds holding the 2 strands together. Two separate strands which are kept separated by the single-stranded binding proteins. I said helicase is like a pair of scissors. Whoops, wrong direction. Here are my scissors, and then here are my single-stranded binding proteins that are actually pulling them apart to keep them single-stranded. That's why we call them single-stranded binding proteins. We want them to keep them apart long enough that we can build the new half of the strand in the middle. Oops. Yeah. DNA polymerase 3 adds nucleotides in the 5 to 3 prime direction. This is our leading strand. So that it will work for the leading strand, which is oriented correctly and is synthesized continuously. I can start, I can go all the way to the finish line. In the lagging strand, it slipped in the wrong direction. It's going the wrong way. Instead of going 5 to 3 prime, it's going 3 to 5. So the DNA polymerase can't work as smoothly on that side. It's synthesized, my lagging strand, in little segments called Okazaki fragments, which are then sealed together by ligase. And we discussed that. I've got RNA primase. I've got DNA polymerase coming in for every little section that is exposed. And then I've got ligase putting those strands together and gluing them in. Mistakes can be made every once in a while during replication, but DNA polymerase 3 usually corrects them. So remember I said that if the wrong partner is there, you either get a bump or you get a dip. So RNA polymerase goes through and it's actually reading. If it comes to a bump, it goes, and it goes back. And when it goes back, it'll actually cut that mistake out and put the right thing in its place. Same with if it runs into a dip, it'll read and go, and go back and then cut out whatever's there and put the right thing in its place. It does have a way to distinguish between the new strand and the parent strand, at least when replication is happening. If this is something that carries over later, probably not. It'll probably be permanent in some way. But initially, when we're going through replication, it's almost like having a proofreader who goes through and looks for misspellings and corrects them for you.